All right, let's talk about some genetic technology. So you should have already done this do now with the carnations and its codominance. So if you are confused, definitely go back and rewatch the video on codominance before you complete it. The learning objective is that students will be able to identify the real world applications of different genetic technology like karyotypes, pedigrees, and cloning. If you don't know those, that's okay. We're going to go over it. And their impact on genetic diseases. So we'll talk about genetic diseases too, which are really, really cool. So genetic technology or biotechnology, karyotyping, pedigrees, and cloning, what are they and what are they used for? So let's talk about the overall field of genetic technology first. So we use genetic technology or biotechnology to help us discover new genes, right? The human genome has been completely typed before, and we can technically write a human genome from scratch. However, we don't know what every single section of our DNA does. We don't even know if all of it has a job. And that's pretty crazy because we've been studying DNA and our genes for so many years now in the field of microbiology, and genetics has existed for a while too. But even to this day, we're still finding new sections of genes and new jobs that genes do. So like I just said, so another use is to discover the purpose of genes. So some genes we have already located. However, we don't know what it does or what the purpose is, or if it's always encoded or when it's encoded. So it's kind of interesting to figure those out slowly as well, or which genes are tied to each other and are important for cohesively working together. We can also use genetic technology to better understand genetic diseases and figure out where the source is, why it's happening, right? During cell division, what's going wrong. And we can also use genetic technology to mass produce chemicals and hormones in a more ethical manner. So before we used to basically, for example, like insulin, people who are diabetic need insulin, and it's going to obviously be produced from a source outside of their own body because their own body doesn't produce it properly or at all. And we used to source it from pigs because they're very similar to us genetically. However, the issue is that it's not ethical to do so because you're basically mass breeding the pigs to produce a lot of insulin and then just taking it from them. So the cooler way to do it now and the more ethical way is to actually use bacteria to do it. Although each bacteria doesn't technically make a lot of insulin at a time, it can reproduce much faster. Bacteria are easy to take care of in a small environment, and we don't consider them as close to sentient beings as us. So it's a lot more ethical and comfortable to use bacteria. But that's only discovered because of genetic technology and the fact that we figured out how to transform DNA and use DNA as the genetic material of life for all living things. So the first technology that we're going to take a quick look at is the karyotype. And if you take a look and you count all of the pairs on the black, red, and green image, you can see that there is 23 pairs. It counts all the way from 1 to 22, and then there's an X. And basically, this is a girl's karyotype. And the reason I know that is because on the X, there are two X's instead of one X and one Y. That would have been delineated otherwise, right? And this is the 46 chromosomes of a human. So there's 23 pairs, so 46 total. And they're paired by the number of chromosome. And like I said before, when you have the pairs together, they're actually the homologous pair, so they match. And one of them came from the mom and one of them came from the dad, and they have the same kinds of genes on them, but they might have different alleles or different versions of the genes on each of them. Okay. So this is where the dominant and recessive is coming in. One has a dominant, maybe one has a recessive, or maybe they both have dominant, or maybe they both have recessives. And each of them encodes many, many genes because they're all coils of DNA. It's not like one chromosome contains one gene. One chromosome can contain upwards of thousands and millions of genes. So the karyotype is actually the process where photos of chromosomes in a cell are taken. So they take a picture of the chromosomes and then they separate them out and splice them to cleanly line them up together and make sure that you have the pairs together and that you can see if there are any extras, any extra sections, any missing sections, any extra whole chromosomes. And it's organized in a way that you can refer to it when you're looking at people's genetic information. And then you can also use it as reference when you're looking at other things. So at the bottom, you can see the example of kind of what it looks like when the chromosomes are just found scattered around in the center of a nucleus. And then you can cut each image apart and put them into their spaces. I actually used to do this in my mom's lab when I was younger, probably when I was like between 10 and 12. I used to work for her lab and just karyotype because kids are a lot better at shape matching than adults are. So this is some cool stuff that is super, super useful in biotechnology. The next thing we'll talk about is a pedigree. So I'll show you a picture example of it on the next page, but if I show it to you, you probably might have seen it before. So a pedigree is a genetic representation of a family tree. So yeah, it's a family tree that traces the inheritance of a trait or a disease. It's only one at a time because it's kind of complex to trace multiple. And they trace it through several generations to see where it pops up, who has it. Maybe you can tell if it's sex linked, if only guys or only if girls have it, or you can tell if it's dominant or recessive based on how frequently it pops up. And it shows the relationships between the family members to help you establish where it's going, why it's going, who has it, where it could be going. And you can also, sometimes they indicate who carries the gene silently. Not a lot of pedigrees actually show the carrying because it's hard to detect to begin with. So you can make assumptions, educated assumptions, based on what the parents have or what the siblings have. So here's an example of a pedigree. This is the black and white one. This is the common one that you'll probably see where, if you notice, all of these spaces or the shapes, the circles and the squares are either completely filled in or completely empty. Some will actually have them half filled to show that it's a carrier or a heterozygote. But these ones, you have to make an assumption based on the information given. So as you can see, there are numbers and Roman numerals on the left side. So 1, 2, 3, 4, these are the generations. So for example, this would be a grandparent generation, parent, and then the current generation, and then their kids. So generations, it's just relative to who is referred to in which level. Okay, the squares are always the men or the males, the circles are always the females. If you have a colored in shape, it just means that you're affected by the condition that we're tracking in the pedigree. A horizontal line between two means that there is mating or marriage or coupling. And then a vertical line down from that horizontal line means that there were offspring made. And we generally do these for human traits and human families, but I guess you could also do this for any other sort of animal as long as they're not ones that make huge amounts of offspring at a time. And the cool thing about this is, for example, I can tell that this is a recessive trait that they're tracking. It's not sex-linked, it's not codominant, it's not incomplete dominant, it's not multiple alleles. And the reason I can tell that is because when, for example, this affected homozygous recessive female marries this man who is not affected, we don't know if he's homozygous dominant or recessive or heterozygous, but we know that he's not affected. However, we can actually make an assumption after we see their kids. And we see that they have 2 kids. And out of 2 kids, when there's a 50% chance, if he's heterozygous, that one of their kids would have been affected, none of them are, which means that he's probably homozygous dominant. But that makes these 2 kids automatically carriers because their mom can only give them a recessive. And that is also proven because when 2 heterozygotes meet, they sometimes will have a kid that has the recessive, because each time they have a kid, there's a 25% chance of that kid getting both of the recessive alleles aside from their heterozygous parents, right? Same case over here. And then here's another similar situation as over here. So the reason why I can tell it's recessive is because it's not like the majority of them have the shaded in. And then also when you track it, you can see that it skips generations sometimes. Okay. It's also not really female male linked either, because if the female has it and it's a sex linked trait, then the boy children always have to have the condition too. So if they're skipping generations, usually you can tell. Here's another cool example. Usually you won't see that many colored in pedigrees, but this one actually shows you the half heterozygous marked individuals. And it's a pretty nice and simple one, but you notice that they didn't write the generation numbers, which is actually a bad thing, because then it's really hard to track for you the number or which individual you're going to refer to. That's the other good thing about this one, is that they have the generation and they have the numbers for each of the individuals. So if you want to refer to a specific individual, you can go generation 1, individual 2, generation 3, individual 10, like that. Okay, and the last one we're going to talk about is cloning. We've actually talked about cloning kind of in vague definition before. So cloning is the process of producing individuals with identical or virtually identical DNA. Either it can be done naturally or artificially. So basically using another organism to produce a specific organism that you use the genes from, or using like basically like a test tube or a simulated environment to raise and develop an embryo. So in nature, many organisms actually produce clones, because that's what asexual reproduction is, right? Bacteria do it, plants do it every other generation. There are ethical implications though, because when you clone animals, for a long time, cloning technology was really, really bad. So animals would die after being cloned, and that was really, really the biggest issue that people had with cloning. However, there are a lot of benefits to cloning. So if it becomes something, and it is slowly becoming something that is a very efficient and reliable procedure, then things like organ transplants, things like running out of food sources will actually no longer be an issue in the future, because we already have too much pollution and we're running out of space on this planet. So it's kind of interesting to think about as a fallback plan if it is something that is done well. And the most famous example of cloning is Dolly the sheep, although we do cloning every day in labs around the country, but it's usually on microscopic organisms like bacteria, algae, things that aren't as ethically implicating for people. So here's a version of cloning where you use a live host. And so you can basically take an egg cell, implant the genes that you want into it, and then put it back into the animal where the egg came from, and have the animal basically birth the original organism that you wanted. And that birth organism will actually have the identical genetic information as the one that you inserted into it, instead of matching the mother. There's also DNA cloning that we technically do all the time in labs. And if you get your PCR test for COVID, not the rapid one, the one that takes 2 or 3 days, that one actually uses something very similar to this in order to be able to see if you have COVID or not. So this is kind of cool, because genetic technology, even in cloning, is used in PCR testing. And so you basically have a bacterial chromosome, and they don't have their little X shapes. They actually have one little circle as their chromosome, and you can insert or take out pieces really, really easily using chemicals and enzymes like cut and paste. And then basically you just have the bacteria multiply over an hour, 2 hours, 3 hours, and they double each time. And then you can get a lot of the same thing at one time after you have that bacteria do its own random natural duplication process. And then you get a lot of the proteins that gene sequence is making, if that's what your goal was, or you get a lot of cells that have this gene in it, and if that's what your goal was. So biotechnology is really, really cool and useful.